Alright folks, gather around. Hey there, wonderful people watching this video. A huge, warm welcome to you all. Seriously, thanks so much for clicking and spending some time here. Before we dive into anything else, sending out the biggest, most positive vibes your way, truly hoping you're feeling fantastic, healthy, and having a brilliant day wherever you are. Science is way more fun when we're all feeling good, right? Okay, let's get into something super cool today. We're going to pull the very blueprint of life, the DNA, right out of a humble strawberry. And guess what? You can totally do this at home with stuff you probably already have lying around. It's not magic, it's science, and it's absolutely mind-blowing to actually see DNA with your own eyes. So, first things first, what is this DNA stuff we're talking about? Imagine DNA as the ultimate instruction manual, the most detailed recipe book, tucked inside almost every single cell of every living thing, including you, me, and this juicy strawberry. It's a long, twisty molecule, kind of like a microscopic ladder that's been twisted into a spiral. That's the famous double helix, and it holds all the codes that tell the cell how to build and run the entire organism. Whether it's making your eyes blue or brown, or telling this strawberry to be sweet and red, that's DNA doing its thing. Now, why a strawberry? Well, strawberries are awesome for this experiment for a couple of reasons. Firstly, they're soft and easy to squish up, which helps us break open the cells. Secondly, and this is the really cool part, strawberry plants are octoploid. That's a fancy science word meaning they have 8 copies of each chromosome in their cells, instead of the 2 copies, diploid, that humans have. Way more DNA copies per cell means way more DNA for us to see when we extract it. It's like trying to find one specific grain of sand on a beach versus finding a whole pile of them. The pile is much easier to spot. Alright, let's talk about what you'll need to become a DNA extraction ninja. This isn't some high-tech lab gear. We're keeping it simple and kitchen-friendly. Grab a couple of fresh strawberries. Frozen ones work too, just thaw them first. You'll need some dish soap. Yep, the regular stuff you wash your plates with. Some salt, plain table salt, is perfect. A resealable plastic bag, like a zip-top freezer bag, is our mixing chamber. A coffee filter or a piece of cheesecloth and a funnel, or even just a strainer, to filter our mixture. A clear glass or cup. Transparency is key, so we can see the magic happen. And finally, the star of the show for making the DNA visible, some very cold rubbing alcohol, isopropyl alcohol. The colder, the better. Pop it in the freezer for 30 minutes or so before you start. Optionally, you might want a wooden skewer, a toothpick, or a small glass stirring rod to help collect the DNA later. That's it. Pretty accessible, right? Now, let's get our hands dirty, or rather sticky. The first step is to prepare our strawberry. Give it a good rinse under cool water to remove any dirt or pesticides, and then carefully remove the green leafy top, the calyx. Pop the strawberry into your plastic bag. Seal it up, getting as much air out as possible. You don't want a strawberry explosion. Now comes the fun part, squishing. Mash that strawberry thoroughly inside the bag. Get your hands in there, squeeze, squish, squelch. You want to completely break it down into a smooth pulp with no big chunks. This physical mashing is crucial because it's breaking open the strawberry cell walls. Think of each cell like a tiny, sturdy house. The outer wall is tough, and inside are smaller rooms, organelles, including the nucleus, the control center where the DNA is kept safe. By squishing the strawberry, we're basically demolishing the outer walls of these cellular houses to get access to the good stuff inside. While you've got your strawberry mush, it's time to make our special extraction mixture. In a small cup or bowl, mix together about half a cup of warm water. Not hot, just comfortably warm, a teaspoon of your salt, and a tablespoon or 2 of that dish soap. Give it a gentle stir until the salt dissolves. Why this weird cocktail? Each ingredient plays a vital role. The warm water helps things dissolve and mix better. The salt? It's not just for flavor. Salt helps break apart protein chains that are wrapped around the DNA inside the cell. Imagine the DNA is like a precious necklace tangled up in a bunch of other necklaces, proteins. Salt helps untangle that mess. And the dish soap? This is the real powerhouse. Remember those cell walls and membranes we broke open by squishing? Well, inside the cell, there are also fatty membranes surrounding the nucleus and other organelles, acting like security doors protecting the DNA. Dish soap is a detergent, and detergents are amazing at dissolving lipids, fats, and oils. So, the soap molecules work like little wrecking balls, poking holes in those fatty membranes and dissolving them, releasing the DNA and other cellular contents into our mixture. It's like dissolving the locks on those inner security doors. Carefully pour this soapy saltwater solution into the bag with your mashed strawberry. Seal the bag tightly again, leaving just a little bit of air inside. Now, gently squeeze and massage the bag for a few minutes. You don't want to make too many bubbles, but you do want to thoroughly mix everything together. We're ensuring that the soap and salt get to work on all the broken cell bits, dissolving those membranes and freeing up as much DNA as possible. Imagine you're gently mixing a batter. Slow and steady wins the race here. Aim for about 5 minutes of mixing. You might see the mixture becoming a bit frothy or foamy. That's the soap doing its job. The liquid should look quite pulpy and cloudy. At this point, inside that cloudy pink liquid, millions of DNA molecules are floating free, mixed in with all sorts of other cellular gunk like proteins, sugars, and broken bits of cell wall. Our next job is to separate the DNA from the soup. This is where the coffee filter or cheesecloth comes in. Set up your funnel over your clear glass or cup. Line the funnel with the coffee filter or a few layers of cheesecloth. Carefully pour the strawberry mush mixture from the bag into the filter. Gravity will do its thing, pulling the liquid through the filter and into the glass below, leaving the big chunks of pulp and seeds trapped in the filter. Be patient. This can take a few minutes. You might need to gently squeeze the filter to get most of the liquid out, but don't force it too hard or you might tear the filter. What you're left with in your glass is a relatively clear, slightly pinkish or cloudy liquid. This filtered liquid is called the lysate. It contains the dissolved components of the cells, including our precious, invisible DNA molecules floating around, way too small to see individually. So, how do we make this hidden treasure visible? Here comes the coolest part, the moment of truth. Take your very cold rubbing alcohol. Tilt your glass slightly. Now, very gently pour the cold alcohol down the side of the glass so that it forms a layer on top of the strawberry liquid. You want to pour it slowly enough that it doesn't mix vigorously, but instead forms a distinct layer, like oil floating on water. Aim for roughly the same volume of alcohol as you have strawberry liquid. The alcohol should sit neatly on top, creating two separate layers, the pinkish strawberry extract on the bottom and the clear, cold alcohol on top. Now, watch closely. Within seconds or minutes, you should start to see something amazing happen right at the boundary where the two liquids meet. You'll see a white, cloudy, stringy, snot-like material beginning to form and rise up into the alcohol layer. That, my friends, is the DNA. You did it. You extracted DNA. Why does pouring cold alcohol on top make the DNA suddenly appear? It's all about solubility. DNA is soluble in water, which made up most of our strawberry extract mixture. However, DNA is not soluble in alcohol, especially cold alcohol. When we add the cold alcohol, it drastically changes the environment around the DNA molecules. It causes the DNA, which was happily dissolved and invisible in the watery strawberry juice, to suddenly clump together or precipitate out of the solution. It's like when you add salt to water. The salt dissolves and disappears. But if you keep adding salt, eventually it can't dissolve anymore and starts to form crystals at the bottom. The alcohol makes the DNA crystallize in a way, except instead of hard crystals, it forms these long, sticky, gooey strands that clump together. The cold temperature helps this process happen faster and makes the precipitated DNA strands less likely to break apart, so they form bigger, more visible clumps. The white, stringy stuff rising up is literally millions and millions of DNA molecules clumping together. It might look like mucus or snot, but it's pure strawberry genetic material. How cool is that? If you want to get a closer look or even collect some, you can carefully dip your wooden skewer or toothpick into the alcohol layer, right into that cloudy white stuff. Gently twirl the skewer around. You should be able to spool the DNA strands onto the skewer, almost like collecting cotton candy. Lift it out slowly, and you'll see a glob of clear, gooey DNA clinging to it. You can touch it. It feels slippery and slimy. This is the actual physical molecule that holds the instructions for building a strawberry plant. Just think about that for a second. You're holding the very essence of what makes a strawberry in your hand. It's pretty incredible to realize that this seemingly simple goo contains incredibly complex information, coded in a sequence of just 4 different chemical building blocks: adenine, thymine, guanine, and cytosine, if you want the science names. This is the same fundamental molecule that's in every living thing on Earth, just arranged in different sequences. So, what did we just do? We broke open the cells by squishing. We broke down the membranes protecting the DNA with the soap. We helped release the DNA from proteins with the salt. We filtered out the big chunks. And finally, we made the invisible DNA visible by causing it to clump together and float up out of the water solution using cold alcohol. This process, in a much more refined and sterile way, is essentially the same basic principle scientists use in labs all over the world to extract DNA for research, medical testing, forensic analysis, and even genetic engineering. They use fancier chemicals and machines, but the core idea, breaking cells, dissolving membranes, and precipitating DNA, remains the same. You just did real molecular biology in your kitchen. Isn't it wild to think that this gooey substance, extracted from a simple fruit, is the same kind of molecule that makes you uniquely you? It connects all life on Earth. Doing this little experiment really brings home the idea that these incredible molecules of life aren't just abstract concepts in textbooks. They're physical things we can interact with and observe directly. It makes the complexity and wonder of biology feel much more tangible. I really hope you give this a try yourself. It's safe, it's fun, it's educational, and the look on someone's face, or your own, when that DNA suddenly appears is absolutely priceless. Grab some strawberries, raid your kitchen cupboard, and become a DNA detective today. Thanks so much for watching, and remember, science isn't just something that happens in labs. It's all around us, waiting to be explored. Stay curious, stay healthy, and keep experimenting. See you next time.